On this week's GARNet Research Roundup, I'm really delighted to be joined by Eirini Kaiserli from the University of Glasgow. And we're going to be talking about a paper just published, open access, in PNAS, the title of which is Zinc-Finger Interactions Mediate Transcriptional Regulation of Hypocotyl Growth in Arabidopsis. So it's great, Eirini, for you to join us today. And if you can give a bit of background and some overview and then some brief details about this paper, that would be great. Thanks so much.
Yes, thank you very much for giving us the opportunity to talk about a recent research and this paper in particular. So, as you mentioned, and based on the title, this paper focuses on the interaction between two proteins that contain zinc-finger domains. But before we go into the specifics of these two proteins and how they work together to regulate hypocotyl growth, I thought it would be important to point out how important light is in regulating development and also plant architecture. Mm-hmm. And although my background is primarily in photobiology and I had worked in the past on photoreceptors themselves, I realized while I was a postdoc in Joanne's lab that it's actually very interesting at the moment to understand how downstream signaling components integrate signals and stimuli, not just from different light wavelengths, but also from different environmental factors and also endogenous stimuli such as hormones, clock, but also temperature and abiotic and biotic factors. Mm-hmm. So this paper focuses on understanding on how a specific signal integrator, so it's a protein that works downstream of the clock and the photoreceptors, how it regulates gene expression in order to regulate hypocotyl elongation, in particular in response to blue light in this case. Mm-hmm. So I should actually thank the person that identified this gene, and it's Olivier Ludet that was a postdoc in Joanne's lab, and now he's at Indra. So he was a geneticist, and he used QTL mapping for isolating this gene that codes for a protein that I'm going to refer to as TZP from now on. Okay. So this protein got its name from its domain composition. So more than half of the protein, or what we say the N-terminus, it's considered to be unstructured or disordered, and towards the C-terminus of the protein there is two zinc fingers, so tandem zinc finger, and a +3 domain, therefore TZP. Okay. So we knew that this protein is involved in regulating plant growth in response to low blue light, and we knew that it regulates a number of genes that are responsible for hypocotyl elongation and plant growth in general, mm-hmm. but we didn't know how it does this. So it doesn't seem to be a canonical transcription factor, although it contains domains such as the zinc finger and the plus that could potentially associate with nucleic acids, but we didn't know how it can regulate gene expression. So one of the first things I did when I was a postdoc in Joanne's lab is go across the street at UCSD, where the K-Lab had just cloned a library of transcription factors, and they were using it initially for yeast-1 hybrid studies. So they were looking for transcription factors that bind to specific promoter elements. But in our case, we used TZP as a bait to screen for the transcription factor library for any transcription factors that could potentially interact with TZP and therefore regulate gene expression. Okay. So from that screen, luckily we didn't get too many candidates because that was our initial figure. We got like 10 candidates, okay. And we decided to focus on the strongest interactor, which is a protein that also contains a zinc finger domain, but also a homeodomain. Mm-hmm. So this protein belongs to a family that is specific in plants. It's the zinc-finger homeodomain transcription factor family, and it contains 14 members.
So you say it's specific in plants, though there's no homology in other organisms at all? It's just plants?
No, so this combination of zinc-finger and homeodomain seems to be specific to plants. Okay. And to our surprise, that there was not much known about these transcription factors. There were a couple of very nice papers from the Irish lab, and they showed that some other members, not the one that we identified to interact with TZP, they're involved in drought tolerance, but there was no information connecting them to light signaling. Okay. So it was interesting to us. So we thought that's something novel. So it would be cool to figure out if these transcription factors also regulate plant development in response to light. Mm-hmm. So that's how the story started. So, of course, we wanted to verify this interaction in a different system, and we could see that these two proteins interact in plants as well, and they co-localize, of course, in the nucleus. And, of course, the next step was to try to see what's the physiological significance. Mm-hmm. So we knew that TZP promotes hypocotyl elongation in response to blue light, and we wanted to see, of course, what's the phenotype of the mutants. Initially, we didn't have a TZP knockout, at least in Colombia, so that was one of the challenges we faced. But eventually, we could find a tDNA line, a SOC line, that was released recently, and we could see the opposite phenotype. So both TZP knockout and ZINC-FINGER HT10, they both show shorter hypocotyl phenotype in response to blue light, and when you overexpress them, you have longer hypocotyls. And then we looked at specific genes, like marker genes that are usually involved in hypocotyl elongation, so specific transcription factors, but also cell wall specific genes. And we could see that these were upregulated when you overexpress these two proteins, whereas the expression of these genes is lower in the mutant background. And then we wanted to focus more on the actual molecular mechanism.
So before we talk about the molecular specifics of this, so you show hypocotyl elongation in the paper that you just mentioned. So do you see any other phenotypes at all with the mutants or overexpression lines? Have you looked at anything else?
Yeah, well, one other phenotype we looked at was flowering, because we had previously shown that TZP interacts with red light receptor phytochrome B, and they seem to be regulating photoperiodic flowering together. Okay. However, we didn't see any obvious phenotypes, at least at the conditions that we were looking at, short days and long days, with respect to ZINC-FINGER HT. Either the overexpressing or the knockout lines didn't have anything obvious.
But that's nice. That's nice, though, right? So you see something which is only specific for the blue light hypocotyl response that isn't involved in flowering. So that's specificity is good in this regard.
I mean, in this case, and that's why we think TZP is actually an interesting protein to work with, is that it seems to be forming protein complexes specifically to either red or blue light, and possibly other wavelengths as well. I mean, there was another recent paper on TZP just published showing that TZP is involved in Phi-A-mediated regulation of hypocotyl elongation. Okay. So we know that TZP is a downstream signaling component and possibly integrates signals not just from different light stimuli, but also from other environmental and endogenous stimuli. Okay. So it is interesting to see specificity. And of course, for the future studies, we should start looking at a tissue-specific level. And of course, it's going to be more difficult, but this will give us, more information in terms of what are these different protein complexes and how TZP is involved in these complexes.
The second half of the paper is more to do with the type of genes which how they interact at genetic loci. So tell us a little bit about the experiments you did to show that.
Yeah, so initially we just did some chromatin immunoprecipitation assays and targeted to specific promoters. So we knew the genes that TZP and ZINC-FINGER HT regulate. So we wanted to see if they associate with the promoters directly, and we could see association. But then, we were not satisfied. We wanted to have a more unbiased approach. So we used chip sequencing, and we wanted to see what does TZP bind to and what does ZINC-FINGER HT bind to in general, and whether there is any overlap. Since these two proteins work together, at least in response to specific light conditions. So we could see there were over 1,000 target genes that were bound by both proteins, and there was an overrepresentation of the binding in terms of promoters, which was quite expected because they're both involved in regulation of gene expression at the transcriptional level. And within these 1,000 targets, there was a huge, again, overrepresentation of genes that are involved in transcription. And when we looked at the motifs, if there was any overrepresentation in the motifs of these promoters that these two proteins bound to, there was the obvious candidates. So light responsive elements such as the G box or the sorolipes, which again, it was a huge relief, and it was another indication that actually this interaction, must be physiologically significant. And these two proteins are involved in regulating gene expression in response to light. And in addition, we could see whether each protein binds to specific targets that they're not common. So for TZP, again, there was a huge overrepresentation of genes that are involved not just in transcription, but also in chromatin remodeling, which was very interesting. And for ZINC-FINGER HT10, we could see that there is some genes involved in drought response, again, which would make sense because other members of this family had been shown to be involved in abiotic stress. And also there were some of the common genes were involved in hormone responses. So again, it would suggest that TZP is involved in integrating signals from different stimuli, not just light, which actually I think is one of the most important aspects and something we need to keep an open mind, to think of the big picture and not just our favorite photoreceptor or pathway, because obviously the plant has to respond to different stimuli at the same time and make a decision whether to grow or stop growing or differentiate. So
that's true. So just to finish, I guess, so that sounds like really perfect actually for kind of future work. Not only have you got a kind of a drought angle, which is nice for kind of translational studies, but also a master regulator of multiple signaling pathways. And I'm sure the interaction studies you've done, with the different genetic loci, there's plenty of things which you can now look for and move forward for, which I won't ask you about now, but we will look out to see those papers in the future, I'm sure. So that's great. So thank you very much, Eirini, for chatting to us, and I say we look forward to speaking to you in the future about all those future papers.
Great, thank you so much. That was great. It was a pleasure talking to you.
Thanks very much. Cheers.